That's where we're starting off. You could call those learning goals. You can call them whatever you want. These are the two big questions that we're going to answer in about the next week. Okay? What is DNA really? And what does it actually do? And it may surprise you. You may not be made of what you think you're made of. You just never know. It's deoxyribonucleic acid. People said it makes you up. Is it your skin? Is it your eyes? Is it your hair? No. What it is a molecule. DNA is a molecule. What is a molecule? Ooh. What's a molecule? We're kind of going backwards through this. So you had physical science last year. Half of it's chemistry, half of it's physics, baby physics. What's a molecule? Okay, small cell. What else we got? What else? Particle, okay. What else? More. Add to that. Particle, little thing. I like those. Is a molecule bigger or smaller than an atom? Bigger, smaller, bigger, smaller, bigger, smaller, bigger, smaller. Bigger. It's bigger. A molecule is made up of atoms. So, and a cell is made up of molecules. So if we go from the tiniest thing out. I'll draw my little ladder rungs here. Okay. DNA is a molecule. So let's talk about a few other molecules. We could have a water molecule. What's a water molecule made of? Hydrogens and oxygen. Sound reasonable? Okay. We have one big oxygen molecule. We have two little tiny hydrogen molecules. They're all hanging out together. Looks something like that. We very often draw water as a Mickey Mouse. It's my favorite way to draw water. It's made up of 3 atoms. It's a tiny little molecule. We could have a molecule of sugar. Okay, sugar molecule. Great. Sugar molecule. What does that look like? Well, we got 1, 2, 3, 4, 5, 6, 7, 8, 9. You don't have to draw this exactly. 10, 11, 12 sugars. See, nope, sorry, 6 sugars. Ha. I drew 2 sugar molecules. 6 sugars, some oxygens, some hydrogens. You do not have to draw this correctly. You don't even have to draw this. C6, H12. We got a bunch of hydrogens, okay? It's a molecule. It's a bunch of individual atoms that are hooked together. That's what a molecule is. That's what a molecule is. Get rid of that. We can have molecules of all kinds of chemicals. Good. So when we say DNA is a molecule, it is. But the first thing you should know is it's huge. It's enormous. Huge molecule. Okay. If we think of a molecule as a building, a garden shed is a building. Little 8x8 garden shed out in the backyard. Keep the lawnmower in there, maybe a ladder, Maybe it's your secret hiding place to go and, you know, you're mad at your sister. I don't know. That's a building. So is this high school. There's a difference of scale. If a water molecule is a garden shed, DNA is this building, this high school. It's enormous. It is fantastically large. You've got miles of it in your body. Really? Miles. You are complicated. Complicated and busy individuals. So it's a huge molecule, and it's not just huge like this building is tall. This building is long. And DNA's size is primarily in its length. Okay, DNA is primarily, it's a long, long molecule. And the thing that I usually equate it to is yarn. Okay, so DNA is this big, it's a big, long molecule. And what makes it, one of the things that makes it special is the way it's built allows it to do some pretty magical things. So, what you have is a ladder. You've all seen a ladder, right? Yes? Who here has not seen a ladder? If you haven't seen a ladder, I'm going to assign you homework. Not a ripple. Okay. Meaning you would have to go home and find a ladder, and that would be your homework. It's only funny inside my head. I understand that. Okay. DNA is basically shaped like an enormously long ladder. If you had a ladder that was miles and miles and miles long, that's what you're looking at with DNA. But those sides, if we look at those sides, they're made up of little tiny atoms all joined together. Various things. You've got carbons and you've got sulfurs and you've got phosphor. You've got all kinds of atoms in there. It doesn't matter what they are. But these sides are made up of specific chemicals. And the rungs are pretty special because actually the way the rungs are built is in halves. What color here? And those halves always match up. Always, always, always. Okay. So the halves are called, the chemicals that make up the halves are called bases. Okay. Those halves are called bases. Halves that make the rungs of a ladder. This is basically like a kindergarten, you know, hold your buddy's hand going to the bathroom kind of thing. Those bases can only match up in a certain way. And if you think of it as like a key and a lock, that's how it's usually shown. So we had our rungs of a ladder, and we have A's that are shaped a certain way. And the only thing that pairs with an A is a T. So we have adenine and thymine. You will never find them paired with anything else in DNA. So that's adenine, thymine. Shrink this a little bit. Is that, and you know, I talked about color contrast and then I went and did that. That's probably not all that easy to see, is it? Let me change the background. Okay, so we've got adenine and thymine. I know, I didn't spell them very well, and I made it smaller to boot, and that's really tough. You'll probably want to get those names out of your notes, and we'll write them bigger in a moment. Or out of your vocab, rather. And the other pair that we've got are guanine. Yes, ma'am. Hey. Okay. Have a seat. Join the fun. Make sure you do the what do you know after class. Are guanine and cytosine. I'll write this one out here. I can do better. Okay, thymine and adenine. So we've got this base pairing rule. They can only line up in one way. They can't ever line up in a different way. Convenient. So what we start to see is that an entire strand of DNA, actually, that long, long, miles-long ladder, really, well, I made that too small. Oops, what color? Wait. Yeah, I did A in red. Actually, let me cut that. If I give you a half a piece of DNA, if I take that ladder and I take a chainsaw and I cut it down the middle, it's pretty easy to fill in the other side, isn't it? It is. Because we can use the base pairing rule to match up what's going to be on the other half of that ladder. So across from A, you always have what? T. Can't have anything else, it won't match up. Across from C, you always have G. Across from G, you always have C. Across from A, you always have T. Across from T, you always have A. Across from C, you always have, I know this is getting repetitive, huh? G, G, C. What comes next? T. Always and forever, holding hands. They cannot line up any other way. If I give you half of a sequence, one side of a piece of DNA, you can automatically fill in the other half. And actually, that will be a quiz question tomorrow. Well, that's convenient. What does that make possible? Anybody think of any special applications for this? What do you think? Fixing DNA? Okay, maybe. We do some DNA repair, not a lot. What do you think, Grant? Splicing DNA? What does it mean to splice something? To mix it with other DNA? Okay. What do you think? To find mutations? Oh, because we could easily see if they were lined up wrong. Okay. What do you think? What? Creating a... Oh, creating more DNA. It'd be really easy to make a new strand if they always line up the same way. What do you think? Creating life? Harder than you think. To go from non-living stuff to living stuff. Tougher than you think. Okay, let's look at let's look at that idea of copying DNA. What do we call it when we copy DNA? It's been a vocabulary term twice now. Replication. I heard it. Replication, that's copying DNA. To replicate, make again. To replicate is to copy DNA. So if we take this strand of DNA, whoops, and if you're doing this in docery, of course, you're getting all the pretty colors. If you're doing it on paper, it's a little tougher. Because what I'm about to do is going to make those of you who are working on paper cry. No, won't make you cry. I don't think. I don't know. Sorry. We're going to take, nope, I didn't get all of them. We're going to take this strand of DNA, and remember, this is like a ladder. Okay, it's like this long ladder. And we're going to take a chainsaw out of that ladder, and we're going to unzip it. Can you do that? How many of you have seen a zipper? You have seen a zipper, right? If you haven't seen a zipper, you have homework. Zippers have teeth that interlock. That's very much what DNA unzips like. It comes apart like a zipper. Well, if this came apart like a zipper, and now we have two halves of DNA, could we make a full piece of DNA from each of the halves? Do we have a pattern here? Well, what would, let's make this new piece of DNA. What would go first? T. Then what? G. Then what? C. Then what? T. Then what? A. A. What next? G. G. What next? C. What next? T. Okay. Put it together and what do you have? Bibbidi bobbidi DNA. Doesn't sound the same, does it? That was easy. It's a pattern for copying itself. It's pretty cool. Can we do the same thing with the other side, or does it only work on one side? It works on both sides. It's about the coolest thing you've ever seen. What goes first? A. What comes next? C. Then what? G. Then what? A. Then what? T. What? T. C. C. G. A. Yay! We made DNA! Again, we sort of push it together. It's a pattern for copying itself. It makes replicating itself really stinking easy. If you can copy letters, you can make DNA. Well, actually, you're making DNA right now. Somewhere in some cell in your body, DNA is being copied at this moment. You got like 30 million cells. Some of them have to be copying their DNA right now. That's DNA replication. It's pretty cool. Let's see. Okay. Right there, we're gonna stop. What I want you to do, and this is something I want you to be in the habit of doing. Every day after we talk about something, I want you to go to your vocab. I want you to look at that part C. Try writing part C in your own words. Today we did word 1, 2, 3 through 6. So that's what we've done today, is words basically 1 through 6. So vocab part C, numbers 1 through 6. Okay, you're going to do that. The other thing that I'm going to have you do, who here likes drawing inside on a rainy day? It feels like indoor recess. Feels like elementary school, and it's very cozy, and could we light some candles and have a cup of tea too? On page 185 in your book is figure 10-1. We got page 186, figure 10-3. Okay, you got page 185, 186, 188. There are 3 pictures there, figure 10-1, 10-3, 10-5. They are pictures in your textbook. Your mission, not should you choose to accept it, I'm just going to make you do it, is to copy those pictures on paper. You do have to do this on paper. I suppose you could draw it on dosseri if you know what, let's not even go there. Let's not even go there. We're not going there. On paper, with a pencil, with your hand. I have colored pencils that you can use. You're going to have the remainder of the time here to work on it. If you don't finish it in class tomorrow, well, it'll be due Monday when you walk in. You'll have another assignment that'll get added tomorrow. I am not going to be here tomorrow. I have to have some stuff done. But tomorrow you'll have this to work on. You'll have a head start. You guys have extra time. And then you will have class time tomorrow to work on it with an additional thing. So do those pictures, and you're going to have a recall quiz tomorrow also. So look over what we've talked about today. We didn't talk about a whole lot. What did we talk about? Did we answer either of our 2 questions? I think we started to answer one of them. What it really is. It's really a long molecule made up of specific things. Okay, tomorrow, recall quiz, this, and one additional thing.